Hello everyone, this is Himanshi Sharma. So today we are going to discuss about signal mediated transport through nuclear pore complex. Here are the list of content we will be discussing about. First, structure of nucleus. Second, nuclear pore complex, nucleoporine proteins, nuclear localization signals, nuclear import and export receptors, role of RAN GTPase in active nuclear transport, nuclear import and export, RAN independent nuclear transport. The major question we will be able to answer after this presentation is that how does signal mediated active transport of macromolecules occur between cytoplasm and nucleus? Before that, we will be having discussion about what is the structure of nucleus. So we will be having discussion on the major components of nucleus, that is outer nuclear membrane, perinuclear space, inner nuclear membrane, nuclear lamina, chromatin, nucleolus and nuclear pores. So let's elaborate each and every component in some more detail. So outer nuclear membrane is phospholipid bilayer and it is continuous with the rough endoplasmic reticulum. Perinuclear space is a space between outer nuclear membrane and inner nuclear membrane and it is continuous with the lumen of the rough endoplasmic reticulum. Inner nuclear membrane, it defines the shape of nucleus. It defines the shape of nucleus and it interacts with the nuclear lamina and chromatin. Nuclear lamina are basically type 5 intermediate filaments and they play a major role in the cell division, DNA replication and RNA synthesis. Now let's discuss about chromatin. In our previous video also we have discussed about chromatin and there are two types of chromatin, euchromatin and heterochromatin. Now nucleolus, this is called the ribosome factory of the cell, and then nuclear pores, outer nuclear membrane and inner nuclear membrane seems to fuse there and it is formed from the nuclear pore complex. Now we will be discussing about nuclear pore complex. So nuclear pore complex is made up of multiple copies of around 30 nucleoporins. There is a membrane embedded ring structure called annulus and surrounding a large aqueous pore and approximately 800 nanometer long filament extend into nucleoplasm with distal ends. These are the distal ends and they are forming a terminal ring structure and called the nuclear basket. And the cytoplasmic filaments, these are the cytoplasmic filaments. They are extending from the cytoplasmic side of nuclear pore complex into the cytoplasm. Now there are like other nucleoporin also, like Fg nucleoporin proteins and structural nucleoporin Y complexes. We will be discussing these in the next slide. Now the total mass of pore complex is around 60,000 to 80,000 kilodalton and the count is 3,000 to 4,000 NPC per cell. Molecules up to 40 kilodalton diffuse passively and macromolecules are transported actively through the nuclear pore complex. Now they're transported out from what are the components that can be transported out from the nucleus to cytoplasm are mRNA, tRNA, ribosome subunits, and what are the components that can be transported in from the cytoplasm to nucleus are histone, transcription factors, and DNA and RNA polymerase. Now moving on to the next. Now we will be having a bit more discussion on nucleoporins. So there are three types of nucleoporins: structural, membrane nucleoporins, and Fg nucleoporins. Now structural nucleoporins, there are a set of 7 structural nucleoporins that form Y complexes. These structures form a Y-like structure. These are the Y complexes, and 16 copies of the Y complex form basic structural scaffold of the pore. Now there are Fg nucleoporin. F stands for phenylalanine, G for glycine, and they form the flexible gel-like matrix. Now this is the D-strom image of NPC and integral membrane protein GP210, which is isolated from Xenopus laevis oocyte. And this B to D structure, they are the fluorescent circular structure. These are the eightfold symmetrical arrangement of the GP210 protein in NPC. And this is the electron microscope image of NPCs. Now let's talk about the nuclear localization signals. They are responsible for the active nuclear import process, and they are rich in positively charged amino acids like lysine and arginine. Example, in the simian virus SP40, NLS is a 7 residue sequence rich in basic amino acids near C terminus of protein. These are proline, lysine, 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 arginine, lysine, and valine. So here are some experiments regarding NLS signals. First is a gold experiment. In this experiment, the NLS signal of nuclear protein was coated with a gold particle and it was injected into the cytosol. And after observing through the electron microscope, it was observed that the particle binds to the tentacles like fibrils and that extend from the rim of NPCs into the cytoplasm, and they proceeded towards the nucleopore complex. Now there is another experiment, that is T antigen mutant in SP40. So there was a mutation being introduced in the NLS signal of protein, and it was observed that the wild form of that protein localized into the nucleus, but the mutated one accumulated in the cytoplasm and wasn't able to pass into the nucleus. Here we can see the lysine is replaced with the threonine, and then the nuclear import was inhibited. Now there is another experiment in which the cytosolic proteins were fused with the nuclear localization signals, which shows that the chimeric pyruvate kinase containing sp40 NLS at its N terminus were expressed in the cell and it was localized in the nucleus. This second figure is showing that the cytoplasmic cytosolic protein has been transported into the nucleus because of attachment of an NLS signal to that. In the first figure, those proteins were present and in the cytoplasm only, but after the introduction of NLS signal to do cytosolic protein, they were moved inside the nucleus. Now let's discuss about nuclear import receptors. So these are the soluble cytoplasmic protein that bind both to the nuclear localization signal present on the cargo protein and also to the NPC protein. Here we can see this is the nuclear import receptor, this is the cargo protein, and this red part is the nuclear localization signal, and the nuclear import receptor is bind to that NLS signal. Now sometimes what happens is that additional adapter proteins they form a bridge between the nuclear import receptor and cargo protein NLS signal, and it is called the adapter protein. Now let's talk about RAN GTPase in nuclear transport. So RAN is a molecular switch that exists in two conformational states. Now one is RAN GTP. RAN is bound to GTP, and the second one is RAN GDP. Now RAN GTP exists inside the nucleus, and this form exists inside the nucleus, and the RAN GTP is present on the cytosolic side. And there are two more proteins. This RAN GAP. RAN GAP is RAN GTPase activating protein, and then RAN GEF. This is guanine exchange factor. Now RAN GAP dephosphorylates the RAN GTP and converts it into RAN GDP, and RAN GEF phosphorylates the GDP and converts it into RAN GTP. Now let's talk about nuclear import and export one by one. So here we should focus on this nuclear import receptor. This is our cargo protein attached with NLS signal. Now this NLS signal is getting bound to the nuclear import receptor. This nuclear import receptor moved inside the nucleus, and then RAN GTP replaces the cargo protein, and then RAN GTP binds to the nuclear import receptor. This complex moves outside from nucleus to cytoplasm, where RAN GTP and 1-phosphate get dissociate from that, and nuclear import receptor is present in its native form again. And this was the nuclear import. Now, looking towards the nuclear export, focus on this again. This is the nuclear export receptor. It is going inside from cytoplasm to nucleus. Now in nucleus, it is binding with both RAN-GTP and protein with nuclear export signals. Now this complex is moving from nucleus to cytoplasm. In the cytoplasm, it is getting dissociated into RAN-GTP plus phosphate and this and cargo protein, and this again nuclear export receptor is again now it's in its native form to get bind again for with another cargo for export. Now let's talk about another category, which is RAN independent active nuclear transport. It is basically occurs in nuclear export of mRNA. Now we'll just focus on this mRNA. These two heterodimeric protein NXF1 and NXT1 complex bind to the mRNA and they form the RNP mRNP complex in the nucleus. Now this complex is being transported from the nucleus toward the cytosol, where this DP5 dissociates these two proteins from the mRNA, and mRNA is being transported in the cytoplasm for the further process like translation. These ribosomes are getting attached to it. Now these protein which got dissociated are again by nuclear import are they are re-imported inside the nucleus for further use. So this was all the discussion of today's video. Goodbye. Thank you very much, and goodbye.